Hello, everyone. Thanks for tuning in to this week's Back to the Basics webinar. Just want to note that all of the webinars will be recorded and uploaded very soon to the BV-BRC. So you will have access to these webinars after they happen. Just takes a little bit of time to get them uploaded. This week we're talking about phylogenetic analysis within the BV-BRC. And before we start, I just want to direct you to follow us on LinkedIn if you don't already. You can use this QR code to get there. So just a little bit of background about me. My name's Anna Capria. I work on the BV-BRC project and I'm employed at the J. Craig Venter Institute. So at the end, I'll go through the institutions that come together to bring the BV-BRC to you. I have a master's from Keck Graduate Institute, and I studied at RIT in Uppsala previously. So just a little bit about me before we get started. Now, a bit of background about the BV-BRC. If you have been to these webinars before, you will have heard this. The BV-BRC is a one-stop shop for data about human pathogenic and related bacteria and viruses in a scalable resource that has both a robust data source and then a bunch of tools to do analysis. Some key features of the BV-BRC is that we support a diverse bacterial and viral communities. We have real-time data processing and uploading of data. The data is managed to support the exponential growth. So see that we have lots of coronavirus sequences, for example, and those continue to grow rapidly as we sequence many, many people with that. Then our genomes, when they're loaded into the BV-BRC, are annotated, so you have that data to make it more searchable and useful for your analysis. Manual curation of metadata as well, and just a bunch of different tools to help support these bacterial and viral communities, and outreach and educational tools like webinars like this. One of the first workshops was on our private workspaces, and that's definitely a tool that I recommend using if you aren't already. So today we're going to talk about a couple services. So to start, I'm going to spend a fair amount of time thinking about data search, because that's really important when you're thinking about phylogenetic analysis. And then our phylogenetic trees, both the viral and protein gene services. So my goal and objective is to describe phylogenetic tools, do a BV-BRC data search, run some phylogenetic tree jobs, and then view and interpret those results from those jobs. So to give you some background on phylogenetic trees, they're also known as evolutionary trees, and it's a graphical representation of evolutionary relationships among species. And phylogenetic trees are predicted. So as a dog lover, I put just an example of the canine evolutionary tree. And phylogenetic trees are a visual tool to help show the evolutionary relationships between organisms, and they can be constructed on genetic, morphological, or behavioral data. In the BV-BRC, it's all genetic data. A typical phylogenetic tree has branches, which you can see as these horizontal lines. The nodes are points where lineages split or diverge from a common ancestor. So this node is splitting from this branch, so separating A, B, C, D from E here, then A and B, and C and D, and then C from D. The leaves are the terminal tips, so those correspond to a specific species or taxon. In the BV-BRC, depending on what your input is, this could correspond to specific sequences or different taxonomy groups. It really depends on your input. The distance between nodes can reflect the degree of differences. So the fact that the distance between A and B versus E and everything else, you can see that there's a longer distance there, and that can reflect how the differences in genetic data or time, how big of a difference it is. Phylogenetic trees are often rooted with a common ancestor, or uprooted, showing without a specific starting point. And we'll talk a little bit more about that as we go. The BV-BRC viral tree service empowers users like yourself to create customized phylogenetic trees from either selected genomes, genes, or proteins. They can be constructed with either nucleotide or protein input sequences, giving you the flexibility with different data types. There's two main groups of tree tools. So we have the fast tree option, which will calculate a large minimum evolution tree using profiles instead of a distance matrix. This is for efficient tree construction. Then we also have two maximum likelihood tree building algorithms, PyML and RaxML. And these algorithms use a substitution model to enhance the accuracy of phylogenetic topologically inferred from nucleotide sequences. So you want to specify settings for these methods to tailor to your analysis for your specific data. This is me again reiterating the point that the data you're bringing in is important when you're constructing phylogenetic trees. So I said some words that if you're not a phylogenetic tree expert, you might not be aware of. So we'll just go through, starting with what is a minimum evolution tree. And the minimum evolution method is a distance-based approach used for inferring phylogenetic relationships. It aims to find the tree that requires the least amount of evolutionary change, so the minimum number of substitutions or changes to explain the observed differences in sequences. There is no best fit model of sequence evolution, and these are based on pairwise genetic distances. So that is what the fast tree uses. More specifically, looking at large minimum evolution trees. So this is something you'd want to use if you have a lot of sequences. Since large data sets are computationally intensive using the traditional methods, large minimum evolution trees are optimized for scalability and speed. So that means that there's some efficiency, sensitivity to data size. We talked about the pairwise distance, and the heuristic approach reduces computational complexity and runtime. And so thinking about runtime, with the BV-BRC services, the size of a genome or whatever you are trying to compare, so if it's a specific protein or a genome, as the size increases, if one run of 10 nucleotides, we'll say, takes you 15 seconds, then if it was 100 times larger, so 1,000 nucleotides, then that would take 100 times as long. So that's important if you're thinking a job that you expect to take a short amount of time, might take a lot longer if your sequences that you're working with are longer. So that's 1-to-1 increase in time. But if you add more sequences, that could really increase the time not aligned 1-to-1 like that. It could take many more hours, for example, as you have more sequences. So just keep that in mind if you're using the BV-BRC to run jobs and they're taking a little bit longer than you expect. And thinking about the PyML and RaxML peak performance, expect high success with fewer than 100 very long sequences, and then between 100 and 1,000 small or medium-length sequences. And the service returns a NUIC file, which can be rendered in Archaeopteryx Tree Viewer in the BV-BRC or downloaded and viewed with other software. So a little bit more information about PyML, the phylogenetic maximum likelihood. It uses the maximum likelihood framework, which is best fit. Has substitution models that you can select, so either general time reversible, Jones, Taylor, and Thornton, Whelan, and Goldman. And this is important to pick the appropriate substitution model and remember which substitution model you chose as you're doing your analysis. PyML also provides branch support values, so bootstrap support, that will indicate the level of confidence in each branch, and that's something that we'll see after we're running these jobs. And you can define settings with various parameters to be able to have more control over what you expect your tree to look like. PhyloMol is known for its speed and efficiency. It's a popular tool for constructing phylogenetic trees, especially with large data sets. RAxML is the randomized accelerated maximum likelihood, and this is also known for its efficiency and speed. Similarly uses the maximum likelihood approach. It's highly parallelized, so it also increases speed for that, and takes advantage of multi-core processors and high-performance commuting clusters for rapid analysis, which is something that's good for you, as all of the computational work of the BV-BRC is done on high-performance computing clusters, and totally free for users. Also has bootstrapping and customization, and sufficient, scalable, and robust. I mentioned that we use the Archaeopteryx tree viewer, and this is a specialized software for visualizing phylogenetic trees. It has a user-friendly interface for exploring and customizing tree displays. It's interactive, customizable, has annotations that you can add or remove depending on what you'd like to see, which will make a lot more sense as we're looking at it, and supports various formats like Newick trees, Nexus, or Phylo-XML, and you can export and save trees from that. As we're building data sets, one thing that will be important to think about is an outgroup. So an outgroup is a more distantly related group of organisms that's a reference point for your tree. And so that can help root the tree and reduce long branch attraction. So basically, the in-group is what you're interested in, and then the out-group is something that is different enough from all of the rest to be able to use for comparison. Finally, I just want to reiterate data inputs. The BV-BRC is a robust data source, and you want thoughtfully integrated data for your tree development, which will allow you to compare things that make sense to be compared against. So thinking about using the BV-BRC as a robust data source, let's jump over to the website. The BV-BRC can be found here at bv-brc.org. And this is our homepage. I'd mentioned earlier that if you were interested in attending one of our future webinars, feel free to click on that in our news and announcements section. And if you don't already have a workspace, you should have a little option at the top that will allow you to create a workspace. Just to orient you a little bit further at the website, the search bar at the top, you have all of the organisms that we support, advanced searches, tools and services. So these are the tools that you can do your analysis with. B means it's for bacteria only. V is viral. If it says either B or V, then that's one that you can use for both. These services can be used with your own data that you can store in your private workspace like a Google Drive folder, or they can be used with data from the BV-BRC datasets. Workspace is where you'll find all of your data. When you create a BV-BRC account, you are given a stock folder of genome group, feature group, and experiment group. Public workspaces is a great way to share information out. And then the BV-BRC workshop one is a workspace that has information from previous workshops. And this will be one of the places that the videos are uploaded. We have a lot of documentation, so the help tab is a good place to catch some of that. And then if you ever need help or you're stuck on something, this contact us under the About tab is a great way to put in any concerns that you might have, and that will be seen by someone from our team and we'll be able to help. At the bottom of the page, you have uploads and jobs. Uploads are showing that you have no active uploads, and here's one way that you could upload data. There's plenty of other places along the site you can upload. If you specify a file type, then it will automatically stop you from being able to select other file types. So if I selected JPEG, I wouldn't be able to select a PDF from my device. And if you're uploading data, that's all private to you, so no worries about privacy, which will be private to you. Then your job status is all of those services that you might be running. See how long it takes for a job to run. You'll submit it, and then sometimes it'll take a couple minutes for it to run. So for example, this job down here, I submitted it at 2:31, but it didn't start running until 2:34. So don't worry if a job doesn't immediately run, but if for some reason you run a job and it's not starting, you can let us know. Occasionally jobs will fail, and if you're not sure why it failed, you can report this issue. So this will automatically pop up when a job has failed. Report issue, and it'll give the information about what happened. And this is similar to that contact us form, just with a bunch of information. Then you can send that on, and someone from the team will be able to help. Let's orient ourselves with the data a bit more. I'm going to be showing you a few different phylogenetic trees. Recently, I traveled to the Florida Keys and I learned about a herpes virus that is leading to tumors on sea turtles. So I made some phylogenetic trees with that data, but just to see if we even had the genomic data for that herpes virus, I did a broad search, so I'm going to walk you through what that looks like. Since I wasn't sure how it got ported over, just did a broad search of all data types. And this will search for sea turtles and herpes virus, which might have been too broad. So we'll just do sea and turtle. And that is searching all fields across the BV-BRC. So here is that herpes virus. We have 211 genomes with that. So this is the Phylogenetic alpha herpes virus 5. And you can see that when I clicked genomes, it just pulled out all of the genomes that were searched for. But there are other ways to kind of refine your data. So I'm going to go back to the search page and only click on one of the genomes. And so then you can see the taxonomy, which we couldn't see before. So now I can click on this Phylogenetic alpha herpes virus 5, and this will give me all of the genomes that we have that are this. So it could be that not all of them were sea turtles, which is true. If you look at the host common name, we have a bunch that aren't labeled, so we're unsure what the host was with those. But this is just one of the ways that you can search for data. Might be interested in further refining your data with filters. And this is all something that we went through over in the first workshop. So once that's posted, I highly recommend looking at that further. But I made a genome group with these genomes. And since we have both a viral genome tree or a gene protein tree, I wanted to see if I could make a genome group with a specific protein. So from just these alpha herpes virus 5, I took all of the DNA polymerases and then I created a group of those. And so you just go new group, feature groups, call it whatever you'd like. And then that will make a feature group because this is proteins, not the genomes. And then if you wanted to go back to the genome view, you can click genomes or genomes at the top. And if I made a group from here, this would be only those 92 genomes that had the DNA polymerase annotated. And then you could make a group of these genomes, just showing you different ways that you can search through the data. So why I decided to do the DNA polymerase for my comparison was that I know that those sequences will all be similar because it's comparing the same thing. And that is something that you just want to keep in mind, especially if you're working with a segmented virus or something a little bit more complicated. You want to be able to have common points that will allow you to make your comparisons. So I had said that I had already run these jobs, so we'll go through how I did that using the viral genome tree. So again, we just went tools and services, viral genome tree. You can bring in either from a genome group, which is what I'll show you now, or aligned FASTAs or unaligned FASTAs. These can be either from a previous job, like maybe you aligned them using the multiple sequence alignment tool that we offer, or you had aligned sequences that you came into the BV-BRC with, whatever the source. These are just some options for how you want to input your data. So I am going to use the folder view to just make sure I'm clicking the right thing. So I'm going to go to genome groups. And since we're in the viral genome tree service, this will only allow me to select genome groups. I'm going to click my sea turtle herpes virus group, and then you press the plus. And because this is an exploratory work for me, I'm not going to select any alignment parameters. And when I ran this for you, I did actually all 3 trees. And I used the GTR model, but you can feel free to use whatever model you would like. I'm going to specify an output folder. One good output folder could be experiments. So then you just click on the little file folder thing, and then you can click whatever folder you'd like. And I'm going to call this sea turtle herpes virus Rex. No. One important thing when you're selecting a phylogenetic tree job on the BV-BRC is to add metadata features that will be able to be colored or searched on your tree. So I'm gonna add collection date, collection year, isolation country, and so you just click on host common name. You click on the feature that you would like to add, and then you can click the little arrow and then it'll move over. And there's no limit to how many additional metadata features from this list you add. It's just limited by what's annotated on your data. So since herpes virus doesn't have this influenza clade information, so technically I could select this, but it's not going to add any value to our data. Once you have all the parameters set, then you hit submit. And then that will put this message about how you're creating a tree and your job has been submitted successfully. You can see now down here, there's one job running currently. All of the BV-BRC services have reference guides, tutorials, and most have videos. So if you wanted to go back to how to run the viral genome tree job after this, the tutorial is a great place to start. And I want to point out the Archaeopteryx reference guide. This has a lot of good information about all the different ways that you can work with the Archaeopteryx viewer, which I'll kind of quickly go through. But if you were like, what is the P meaning? Or what do the different zoom settings mean? This is a great place to just align yourself with how to become more comfortable with Archaeopteryx. Going back to our jobs, you'll see that it's running in the viral genome tree service. The gene tree is very similar. It just uses either DNA or protein as an input on a feature group or aligned FASTAs. But similar parameters. The models are a bit different, but the metadata options are there as well. And this is really just thinking about what data you're bringing in. Now I want to go through the jobs that I've already run, the FastTree, PyML, and the RaxML. Once a job is complete, you can get it from this job view, and you just double click, and that will bring you to the output of the job. You can see that the file path at the top were in the experiment groups, so you can also get there by going to your workspace, experiment groups, and get the information. If you weren't sure what parameters you originally ran, just checking the parameters is a great way to orient yourself that you were using the viral genomes resource, using the file ML with the GTR substitution method, and so on. So that's a good way to just remind yourself of your inputs. At the top right of every job, you'll have this view icon, and for the phylogenetic trees, this will pull you to the Archaeopteryx view. But you can also get there by clicking on the new tree or the file.xml. This job will align your sequences. You can get that align file and also a report. But let's look at the view of this, the sea turtle genomes. I'm glad this came up. We have a bit of a tiny bug that sometimes doesn't load Archaeopteryx on the first try. So if you're like, I'm supposed to see something here and you don't see anything, I would just reload your screen and then your tree should load. Here is our tree, but it's not that clean. So this is when it's important to think about how to make it make sense for your view. So there are some things that we can do right off the bat, like remove the confidence. Now, if you need the confidence for your journal article or whatever, it's important to know that there are different confidence levels on the branches, but how it is right now makes the view a little bit messy. So you can remove that, for example. Right now, the nodes are labeled with the genome name. Could have the node name added. This is assigned through the service, so not super helpful having a random number. Can have the branch lengths added or removed. You can change the size of the labels. You can zoom in and out, so zooming up on the y-axis, can zoom on the x-axis, which kind of declutters some of these areas, for example. And then some additional visualization tools through labeling. So if you remember when we had put those metadata features in, we could label based on collection year. Doesn't seem like there's any strong group based on the collection year. Could label based on geographic location. So here you can sort of see this virus, depending on where it's isolated from, there is some grouping. So like those from Brazil are up here at the top, the ones from Australia, Bowen grouped together, and so on. So this is sort of starting to make sense of this output. You can do that with the different colors labeling. And the default is black. Can also label the nodes themselves. There are some features that aren't super helpful, like genome name or genome ID. That's going to pull you out a bunch of different, since each one is different, that's not a helpful color scheme. So just keep that in mind as you're labeling by color. You can also change the shape of the nodes, and you can move these bars around so that you kind of get a sense of what it's going to look like. So for this one, adding the node shape, I'm going to make the node fill the geographic group. Then this default can also scroll in, out, can make the nodes bigger. And so something like this, it's not super helpful to have the different shapes. A smaller subsection of the tree would give you a better view for that. But just kind of showing you the capabilities. And so if this is what I wanted, which I don't think that this would be a very nice image, but for the sake of just showing you, you could download this and it will basically be like a screenshot. It will download exactly what you see except for the guides like this. So it's basically, in this case, just this information. So you can scroll and move it over, so maybe you want it closer to your label or whatever. Just showing you that you have the flexibility to move stuff around, which is valuable. So that was the genome version. I'm going to show you the DNA polymerase one. So we'll look at the fast tree version of that and view that job. This is a lot more linear. You can see I made an outgroup with a hepatitis B virus DNA polymerase and a little bit isolation country. You can see that moved pretty similar. There's a lot of Brazilian sequences here, so just showing you that you get the same view from either the viral genome tree or the gene protein tree. That's a very quick overview of the Archaeopteryx output. Can see this is the job output, but it doesn't quite look the same when you're clicking through from here. So the icons mean something on items on the BV-BRC. So if you click on the flag, that will bring you to the jobs page, which has the parameters and just this view. So just know that there are ways to get back to there. It'll make you a tree as an SVG image automatically. This tree is not super helpful as the labels are not corresponding very much, so I'd recommend going into Archaeopteryx mode. The tree report, that will show you the tree. It showed where the data came from, how it was aligned, how it was used to build the tree, and how Fig Tree was used for the SVG. And so if something went wrong, the gene tree report would explain where things went wrong. Can download this HTML file, can get the tree in Newick form, and this will open up Archaeopteryx so that error message will come up. I've already seen it. Go back here. The other output is log. This is the log of the tree. Job has some of that information like model of substitution, just more of the math behind this predicted tree. And the citations are here as well. So I'm just going to hop back over to the presentation. We talked about these outputs just now, but you have the aligned FASTA format, so it is running in alignment. Then the Phylo.xml format, Newick format, stats.txt. That's what we just looked at with the statistics. And then the tree report, which we looked at before that. So to recap this very quick introduction to the BV-BRC, gave you some background on phylogenetic trees, description of the BV-BRC tools, how to run the jobs in the BV-BRC, and then the standard outputs. The BV-BRC project is a combination of 4 institutes. So we have the University of Chicago, JCVI, University of Virginia, University of Alabama, and you're funded by NIAID. It's a bioinformatic resource center. So now I'll leave it open for any questions that you might have, but I really appreciate you all joining me today and look forward to seeing you at our next workshop. So the visualizations box, if I didn't already answer that, when you view the Archaeopteryx tree from the job, then the visualizations box will be in the top right. So if you're not seeing it, I would go back from the jobs view. So click on jobs in the bottom, click on the row, and then this view icon. That'll be the best way to make sure that you see the Archaeopteryx tree. The question about your view being saved, it does have some memory, so I can't promise that it'll be perfectly how you saved it, but it's kind of like it's cached with the latest settings that you used. Like before we started, I moved out the x-axis a lot, and that's why the x-axis looked pretty big on all of them. Before I'd done that, they all started pretty flat, so it does save some things like that. So you color the text under the visualizations. And so that's why it's really important when you're running this job that you add those metadata features. So I'll go back to that in a second just to make sure that hits home, because it's a real power of adding value to your visualization. So since I added the metadata features and know that this data set has different geographic groups, for example, adding that will color based on the geographic groups from the original files that we used. So these are all based on the information that when you're originally running the job, you click on metadata options and you move things like collection year over, geographic location over. And you're not going to always need these. Like maybe you're looking at a specific region of a country, then the geographic location isn't going to matter. But you're looking at over time, then you want the collection year. Can also remove ones that aren't needed, but it's not super taxing. So I don't think it's that important to remove. I'm gonna get back to you about an answer about mutations if we're making a virus evolution phylogenetic tree. I don't believe that's currently something that we support, but I don't want to say that with complete certainty, so I'll get back to my team and reach back out to you to answer that question. If you're interested in rabies virus, I'm happy to quickly show you how to make a data set for that. I'm a big fan of the global search bar at the top because it's a great opportunity to get a feel for how that data is sorted. But you could also, if you knew the viral family from here or on the homepage, there's some other ways to search, but it should get you down to the same information. So I'm going to go from the taxon view. So these are all the Lisa virus rabies genomes that we have. Then this overview page is a great resource to orient you with what the data is. But we'll go to genomes. And theoretically, you could make a group with all of these right to start, up to 10,000 into a group. But I'd recommend filtering it down based on what you're interested in. So maybe a collection year or comparing country, comparing hosts, all of these different ways to refine the data based on your needs. You can, like if we were interested in only ones from North America and not interested in humans or whatever it might be. So it's hard for me to make a data set without set parameters that you're interested in. You can then just make a group and do your analysis further in there. Hopefully that is enough, but if you need more help with that, I'm happy to help you set up a data set based on your needs, or if you're going to filter out different things and it's not going how you expect, always fill out the contact us. I'm happy to jump on a call. Most of our genomes come from GenBank, and we just organize the metadata. We do GenBank submissions of sequences for influenza virus, which is a great plug for our next webinar, so feel free to join us next week. That's kind of all I've got for presentation, but thanks everybody for joining us.